All right, thank you everybody. Thanks for coming along. It's my great pleasure today to welcome Duong, who has been working in my lab now for the last just a little over 3 years. So Duong's background, she started her undergraduate program at La Trobe in 2005. And then, like a number of very good students, we were fortunate to woo her across the road from La Trobe Bandura to the Bandura Medicinal Chemistry Labs, where she did honours with Jonathan Bale in antimalarials. Short after that, then Duong came and worked at WEHI as an RA for a couple of years with Guillaume on some viral BCL2 protein inhibitors. And then in, just over 3 years ago, joined my lab to begin this project. So Duong's asked that I don't go into too much detail, but I would like to say that Duong has always applied herself incredibly diligently to what has been at times quite a difficult project. Not only always working hard, always coming up with good ideas, challenging my ideas and coming up with better ones, which I guess I had to wear on the chin a number of times. But she's always done that with a smile, and I think, as you'll see today, done some really nice work. So Duong, I'll hand it over to you.
Thank you. So, thanks, Chris, for the introduction. Good afternoon, everyone. So today I'm going to talk to you about my PhD project, which is on the synthesis of Rocaglamide analogues that's targeting the translation initiation factor eIF4A. It's not running. Yeah. So I'm sure at WEHI we've heard a lot of great stories on the development of small molecules, that starting from fragment-based screening or computer-aided drug designs, and it is really great to be able to build a drug from scratch. However, there's another wonderful resource where we can find great drug candidates that's come from nature. And my project is also inspired from a natural product, and it is called Silvestrol. Structure is shown here. It was isolated from the plant Acknowledgement violata that was found abundant in tropical forests in Southeast Asia. And Silvestrol belongs to a bigger class of compound called flavonoids, and Silvestrol, together with its other derivatives, share the cyclopentabenzofuran core structure as highlighted here. So why should we be excited about Silvestrol? It was originally found to be equipotent to, if you know, the famous Taxol or paclitaxel in several human cancer cell lines in solid tumors, such as lung cancer, prostate cancer, and human breast cancer. Later, it was found to be also show efficacy against hematological cancer, as shown here, just a snapshot in patients with patient blood cells with chronic lymphocytic leukemia. As you can see that while Silvestrol is not as cytotoxic towards normal blood cell as it is to the patient whole blood samples, what's even more interesting is that Silvestrol is selective towards B-cell cancer versus T-cell. The importance of this is when you want to treat a patient with B-cell cancer, you don't want to compromise the immune system, and Silvestrol does exactly that because it doesn't affect the T-cells. So the in vitro results also translated to the in vivo xenograft model in ALL or acute lymphoblastic lymphoma, where you can see that treating the mice with Silvestrol prolonged their survival and show no toxicity was observed. At the molecular level, Silvestrol was found to induce a reduction of protein expression of a few different proteins, among which is the MCL1 protein, which you probably recall that is one of the oncoproteins that overexpress in a number of cancer. So because of the balance in protein translation and protein degradation is very important in the life of a cell, That is why inhibiting protein translation becomes an interesting therapeutic avenue. So before the cell commits to undergo translation, this process is regulated by this complex called the eIF 4F initiation complex that makes up of these main components as shown here. So these are the three subunits, including the eIF 4A, 4E, and 4G. So within the cell, there's a lot of convergent pathways to regulate this process, and because it is the initial rate-limiting step, that is why it's an attractive target to develop small molecules to inhibit this. And Silvestrol is among a few examples of natural products that inhibits the eIF4A subunit. Treatment of the cells with these natural products has been shown to cause translation inhibition and resulting in either cytostatic or cytotoxic to the cell. So I'm going to briefly show you how it works. This is just my cartoon illustration of the mode of action of Silvestrol, how it inhibits the cap-dependent translation process. So this is a very simple version of a normal structure of an mRNA with a 5' cap and a highly structured 5' untranslated region. So at the start of translation, you have the cap-binding protein for E subunit in blue binds to the mRNA, followed by the recruitment of 4G and 4A subunits. These together make up the eIF4F complex. What eIF4A does is it unwinds the highly structured 5' untranslated region that allows the whole complex to scan along the mRNA and followed by the binding of ribosomal subunit, which actually carry out the translation process. However, in the presence of small molecules such as Silvestrol, it's been found to bind to the eIF4A subunit and induce a cross-linking of other eIF4As and prevents the binding of ribosomal subunits and thereby shut down translation. So although the interactions of Silvestrol and eIF4A has been confirmed by pulled-out assay or other biochemical assays, its actual binding site is still unknown. And only until recently we were able to obtain some hint on where the compound may bind. So this is a recent study on the yeast equivalent of eIF4A. And as you can see that it overlays quite well with the mammalian eIF4A. And what they found when they did the random mutagenesis on the yeast eIF4A is that there are 6 important residues that contributes to the bindings of Silvestrol or rocaglates. And these 6 residues seems to be close to each other and close to where the binding site where the protein binds to mRNA, which may gives you the thinking that it supports the fact that eIF4A induced the stronger binding of Silvestrol induced the binding of eIF4A and mRNA and shut down translation. So I hope I've convinced you that we have a potentially really good molecule to be an anti-cancer drug that, as I told you before, it was isolated from plants. So you probably think that we can get it, just grow it in the gardens and it's that simple, but is it? It was actually isolated from fruit, twigs, or leaves from this plant, but the yield was very, very low. And in addition to that, Silvestrol has other challenges. It's got a very complex structure, and currently there are only 2 total syntheses of this compound, and it's quite complex to get a good yield to be further studied in biological assays. In addition to that, Silvestrol also has the pharmacokinetic properties challenges, which contribute to this sugar-like moiety in blue, because it subjects the compound to be effluxed out of the cell via an efflux pump mechanism, so preventing it from getting to the target. And also this ester functionality, as highlighted in purple here, is also a weak metabolic spot of the compound. Existing literature has tried to improve these limitations in the compound by verifying this sugar-like moiety. They were able to improve the efflux ratio, and also changing this ester group also improved the metabolic stability. Further good news is that the literature has shown that by replacing the whole sugar moiety with simple substituents still maintain the activity of the compound, and whereas this part of the compound, the interactions seems to be quite important for the activity. However, having improved all these properties of Silvestrol, there's still the narrow therapeutic windows that still remains as a challenge. The previous work from our group has shown that replacing the whole sugar moiety with these simple substituents such as the benzoyloxy or other heterocycle-containing moiety still retain the activity, and this work has recently been published. So, as I've shown you before, that a lot of the focus now has been on this area of the compound, so just verifying the residues the substituents around this cyclopentabenzofuran core. So for my work, I want to investigate the importance of this C8 methoxy group, because it hasn't been reported before, the importance of this in the activity of the compound. And secondly, because current synthesis only allows changing substitutions around this area, what we want to do is to actually modify this core in the middle here via an alternative approach using the Pauson-Khand reaction, which I'm going to talk to you about in a bit later. So that brings me to the first part of my talk, which is synthesize the simplified analogues of Silvestrol without the methoxy group. So what we want to do here is to, based on the previous observation, where replacing the sugar with this benzoyloxy compound retain the activity, what we want to do is to start with that compound, but in order to investigate whether the methoxy is important, we want to make a deletion analog, so converting the methoxy to just simple hydrogen to see whether that affects the activity of the compound. And if you recall, the interaction at this position is important, so we also want to vary the substituents at this position as well. So there's a lot of synthesis of this core out there, but the Paucos approach, which was developed by Paucos group, using the cycloaddition as the key step, is one of the most popular way to generate analogues. And the sugar moiety was developed in the Rizakas group at Melbourne Uni, as allowed the total synthesis of Silvestrol to be carried out. However, for our work, we'll just focus on generating analogues from this core and using the Paucos approach, which involves the cycloaddition of these two intermediates. And if for our work, we want to make compounds without this methoxy, which means that we required to make this key intermediate here, which is called the 3-hydroxyflavone. So there's a number of ways to make 3-hydroxyflavone. For our work, we want to work on a classical reaction, which is quite simple to access the 3-hydroxyflavone from this charcoal here, and it is called the Algar-Flynn-Oyamada reaction. And this reaction has been around for 80 years. The limitation of this reaction is that the reaction wouldn't work if you have an alk substitution that is not hydrogen at this position here next to the carbonyl group. The reaction works via two-step. So it's first is the cyclization under basic peroxide, followed to get to this intermediate, followed by an oxidation to introduce the double bond and get to the 3-hydroxyflavone intermediate that we want. Using this synthetic route, I was able to generate a number of 3-hydroxyflavone. So this is a two-step sequence, starting from this compound here, where you can introduce different R groups to make the charcoal under the presence of base. The first step proceed with no problem. We were able to isolate these charcoals using just recrystallization. And we were able to introduce different R group. As you can see here, these are the more electron-rich charcoals, whereas these ones are the electron-poor ones. However, subjecting this charcoal to the basic peroxide standard condition for the AFO reaction, although we were able to isolate the 3-hydroxyflavone as a desired product, for substrate with substituents that's considered electron-rich. However, when you move to even just without the electron donating group at this position, we move the methoxy to this position, the reaction didn't work. And also, by replacing with the electron withdrawing group, we didn't isolate any product. So the question we asked here is that, was it the electronic factor? So you can see here that electron-rich is more favored than the electron-poor ones. Or is it the solubility of the intermediate, as we observe during the course of this reaction, that there's precipitation formed during the reaction? So from that thinking, we want to optimize the reaction further by starting from this dimethoxy compound. So subjected to this standard condition of the AFO, but what we vary here is the addition of phase transfer catalyst, which is known to be used in circumstances where you have like a biphasic reaction. So the condition here, where it's under standard condition of the AFO reaction, the yield we got for this 3-hydroxyflavone is 19%. However, as you can see that as we added phase transfer catalyst, either ammonium-based phase transfer catalyst or phosphonium-based phase transfer catalyst, we were able to improve the yield of the reaction. And what's interesting here is we're probably observing the counter-ion effect as you move from chloro to iodo, and the yield was increased for this specific substrate. And we probably observe the trend also as you increase the aliphatic side chain of the phase transfer, it seems to compromise the activity of the phase transfer. So that's the dimethoxy compound. What about the compound that with only one methoxy group? As you can see that the same condition does not really apply. So as shown here again, under the using other cold solvent didn't help the reaction, only until we add with higher equivalent of base and peroxide, but in the presence of the phosphonium-based phase transfer catalyst, only until then we were able to isolate clean compound in moderate yield of the desired product. So that's for the compounds with electron-rich substituents. So the more challenging one is the one with electron-poor donating group, this bromo here. So as you can see that while other phase transfer catalyst didn't work and instead give me an unknown product, only this phosphonium-based phase transfer here, with that in the reaction, I was able to isolate clean compound of the one with the para-bromo substituents. So by using phase transfer catalyst, I was able to generate a few analogues of the 3-hydroxyflavone, which was then subjected to the Poco's approach to synthesize the final compound. I'm not going to go through this synthesis, but this is a really elegant way to make this sort of compound based on a biosynthetic pathway of Rocaglamide. So using this sequence of synthesis developed by Poco, we were able to make a few compounds that doesn't have the methoxy group and vary at this position here. So the moment of truth is when you subjected these to the in vitro translational assays to see whether the compound without the methoxy group still inhibit protein translation. So as you can see here, in the in vivo translational assays, we observed that at 50 micromolar, there was no inhibition of the compound of this series, which confirms that this methoxy group here is required for the inhibitory activity of Silvestrol on translation. So to summarize this part, I've talked to you about the synthesis of the analogues without the C8 methoxy here, and the use of phase transfer catalyst has enabled to generate compounds that was previously challenging to make because of their either electronic or solubility nature. So we reported that the use of quaternary phosphonium-based phase transfer improved this reaction, and the advantage here is that using this way to make 3-hydroxyflavone, you can purify it without using column, which cause degradation of this compound. And testing of these analogues with the methoxy shows that it is required for the activity of the compound. So that brings me to the second part of my talk. What I've shown you before is that using a previous approach developed by Poco, we were able to investigate the importance of the C8 methoxy substituents. However, this method doesn't allow you to actually modify the core, and that will be the focus of the next part of my talk. So what I'm going to talk to you about here is our effort to go and try modify this core using the reaction called the Pauson-Khand reaction. So the rationale behind this is, so I've shown here is a simplified structure of rocaglates. I've removed all the substituents, and we think that we will be able to obtain this compound via this simpler precursor, and this, can be obtained from the Pauson-Khand reaction, which joined the bond between an alkyne and alkene and a 1-carbon subunit. So this intermediate is called the E9 precursor. So what I'm going to do here is that's the E9 precursor that we want to focus on. So we think that at first we can try and generate the core structure that's resemble the ones that actually in the natural product. And this will start from a very different double bond to the standard one that participate in the Pauson-Khand reaction. Secondly, we want to vary the ring size of the core using this reaction, so you can extend this to a larger ring size, or also replace the oxygen here with other atoms such as carbon or nitrogen. Ultimately, what we want to do is to try this reaction on a tri-substituted alkenes, because as you remember from the the natural product compound is that both of these positions are substituted. So just a bit of background on the Pauson-Khand reaction. It was first developed by Pauson and Khand, which Khand is the student of Professor Pauson, and I read somewhere that Khand, after finish his PhD, he went and teach at high school, but late at night he would insist on coming back to the lab and just work on a certain project. And that's when he discovered the Pauson-Khand reaction. So what it is it's a 2 + 1 cycle addition. By that, it means it's the addition of 2 carbon, and 1 carbon monoxide unit. And this is called an alkyne, alkene, and carbon monoxide. And there's a lot has been done since its discovery in the 70s. But what I'm summarizing here is very simplified of what has been done. So you form, from joining this alkyne, alkene, carbon monoxide subunit, the product of the Pauson-Khand reaction is this cyclopentenone. This process is catalyzed by metal, and there's a lot of metals studies on different metals catalysis for this reaction. Molecular sieves has been found to promote this reaction, and it's been widely used. And also there's other additives that has been studied to promote this reaction. For my project, we started with the use of this hexacobalt monoxide catalyst that was originally used in the first discovery of this reaction. So the Pauson-Khand can be intramolecular or intermolecular. Intramolecular is when both of this unit, the alkyne and the alkene, are on the same molecule. So this is a simplified version of how the Pauson-Khand reaction works. And it basically has 3 important steps. Firstly is the complex of the catalyst to the alkyne, followed by the loss of 1 carbon monoxide subunit, which is then allows the alkene to bind for the alkene insertion to occur, and then the reaction forwards to form the cyclopentenone with the insertion of 1 carbon monoxide. So within this reaction, there's a lot of studies around adding different additives to drive the reaction forwards, which is what essentially I tried to do. And so I will go through with you here the scope of the Pauson-Khand reaction that I investigated. So at first we start as a proof of concept. We want to start with a simpler reaction. So you can see here we were able to, hypothetically, we were able to make this 6,6,5 ring joined together from this intermediate here. That was the simpler reaction. However, the actual ring we want to do is the 6,5,5 tricycle here, which resemble what's present in the natural product. And this will start from this special alkene here, which is called the enol ether. And then I'll follow by investigating the changing the electronics of this alkene, how that affects the reaction. So first thing first, the most simple one. So this is the ring tricycles that we want to get to. So it's a 6,6,5 systems. So to start with, we generated this sort of intermediate and subjected that to the quite standard condition of the Pauson-Khand in addition of the N-oxide, which has been shown to promote the reaction. And we found that we were able to generate this 6,6,5 ring system quite easily as a 1 diastereomer. However, the challenge is to generate the actual 6,5,5 ring system that's present in the natural product. And in order to do that, the intermediate that we propose is to go via this E9 precursor here. So as you can see that by moving the electron, by moving the oxygen, now it's adjacent to the double bond. And in order to make this intermediate, we go via using the TEPI reagent to convert this carbon-oxygen bond to the carbon-carbon bond here, and subjecting that to a lot of different conditions and varying the conditions, and we couldn't get to the desired product. Instead, the major compound that was isolated out is where we've lost this alkene here, because this is quite a labile moiety. So we asked the questions why moving the oxygen to adjacent to the carbon-carbon double bond doesn't work. It could be the bond distance between this alkene and the alkyne is not enough for the alkene to reach over and form the bond, or it could be the stability of this compound, of this actual functionality, or it could be the unreactive alkenes. And in order to tease out what was causing the problem, we turned to this whole carbon skeleton here. As you can see, we want to replace the oxygen with now the carbon. So that brings me to this part here, where we want to work on the system with just carbon and no oxygen at this position. And we want to investigate how it works on a disubstituted alkene and also the trisubstituted alkene. So first, the disubstituted alkene. So this is the ring system we want to get to, where R1 or R2 is hydrogen. And so in order to do that, the intermediate that we want to work on is as shown here. It can be made from a two-step sequence by first the coupling reaction to generate this, to install these R groups as we wanted, and followed by converting this carbonyl group using the Bestman-O'Hara reagent to convert it to the alkyne. And this route has enabled us to generate a few different enine precursors to work on. And we did the optimization on this substrate here. As you can see that in the presence of trimethylamine N-oxide, we were able to get to a reasonable amount of yield. And so this condition will be used on the other analogues that was made. And under this condition, we were able to isolate this compound as one diastereomers in the presence of trimethyl N-oxide. So as you can see that we were able to make the compound with carbon skeleton instead of the oxygen here. And this is on the disubstituted alkene, where R2 or R3 is hydrogen. So what we wanted to do ultimately is to have a system that can form the one that's trisubstituted, so where both R1 and R2 are not hydrogen. And this is very challenging because it hasn't been studied before in the Pauson-Khand reaction, and this is what we want to optimize this reaction further. So as before, I need to get my enine precursor intermediate, which is also obtained from a two-step sequence. However, disappointingly, subjecting that to the condition that worked before, as you recall, it doesn't give me any of the product. In fact, we get back this complex here. So if you're going back to the mechanism, you would think that these two steps would be where you look at to try and optimize the reaction, and this is what I try to do. So the first one is we want to promote the loss of the carbonyl so that to create this space here, the vacant orbital, for the alkyne to be inserted. So this step, the loss of carbon monoxide, is a very energy demanding, and this is why different physically promoting the loss of carbon monoxide or by adding other chemicals to promote this reaction is able to overcome this hurdle. So what we found that in the presence of N-methylmorpholine oxide, we were able to achieve some product only under thermal conditions, so at high temperature. Other effort to physically promote this loss of carbon monoxide, either using irradiation or adding other like silica or sealite, didn't really improve the yield. So we turn to this second step, the alkene insertion. So because we want to drive the reaction forward, and you would want to add something in this step here to promote the formation of this bond. And studies have shown that the use of Lewis base has been shown to stabilize this complex and helps the reaction forwards instead of going backward to this intermediate here. So what the stabilizing ligand does is it lowers the energy of the formation and allow the reaction to go to the product. So we started out by testing out different Lewis bases, such as the one that containing sulfur and phosphide, but it didn't, as you've seen here, that it doesn't really give us the desired product. Even though with dimethoxyethane that has been shown in the literature to promote the Pauson-Khand reaction didn't work in this case. We then investigated the presence of other alcohol, and we was really excited to see that we were able to see some yield of the desired compound. So as you can see here that benzyl alcohol, this compound here, and also another aromatic containing alcohol was able to improve the yield to not high, but it's quite workable. So we were very excited by that because no one has done this before. So we want to see whether we can improve this reaction further. And by screening other alcohol ligand, we found that even though changing the length of this carbon here didn't to this 2 carbon, it doesn't improve the yield, but you actually lose the yield as you increase this further. In addition to the aromatic containing alcohol, we also screen other alcohol, but until we get to this tertiary alcohol here, that is when we get the best performing ligand. And whereas with the other secondary alcohol, one of the side reactions that we observed was the oxidation of this alcohol under the reaction condition. So having found a ligand that actually works quite well for a reaction that hasn't been done before, we want to see whether we can tease out what was it about the ligand that actually helps the reaction. So as you can see that compared to this aromatic alcohol here, we tested on the one without the aromatic containing alcohol. The yield was compromised by almost a half. And interestingly, when we tried to promote the reaction, so instead of using toluene as a solvent, we moved to a non-aromatic solvent, which is dioxane. The yield was increased, and the ratio of these 2 compounds, diastereomer that we get here, is actually reversed. It favors this number, diastereomer number 2 here, which is the actual compound that we want. And cyclohexanol, interestingly, also give a similar result as observed when dioxane was used as a solvent. So just to summarize on that, we found that this tertiary alcohol that's aromatic performs the best compared to the ones that are non-aromatic alcohol ligand. So in an effort to explain why we observed what we observed, even though we don't know what the alcohol ligand, how it works. But what I can propose here is, in addition to performing as a normal Lewis acid ligand, which stabilizes the complex, the alcohol that contain an aromatic ring is probably performs better than the others because of these secondary interactions, the pi-pi stacking, which further stabilize this intermediate. So that was the discovery of a substitute of a Pauson-Khand reaction on a tri-substituted alkene. But all the example I showed you before, we didn't have these substituents here. And if you recall from the first part of my talk, this methoxy is important for the activity of the natural product. So that is why we want to see whether the Pauson-Khand reactions still enable us to access this sort of compound where we have methoxy substituents such as at this position here or 1 carbon away from the carbonyl. So in order to do that, the intermediate that we need to get to is shown here. So as you can see now, it's substituted at this position or the meta position here. And also the alkene is also tri-substituted. There was a bit of a problem trying to get to this intermediate because of their electron-rich nature. However, by changing slightly the synthesis, we were able to get to these 2 intermediates. And the moment of truth is when we subjected that to the Pauson-Khand condition that was developed before. To cut a long story short, unfortunately, it doesn't work when you have electron substituents on this side of the ring. The compound that we recovered was just the complex of the catalyst and the intermediate with the recovery of the alcohol ligand. So in order to tease out whether it was the electronic factor, so it works when it doesn't have any substituents, but when you put substituents here, it suddenly doesn't work anymore. So we want to try to change the electronic of this alkene by going to the enone. So you introduce an electron withdrawing group to make this alkene now electron poor. And also we want to investigate whether, if you recall, the alcohol ligand was one of the best performing ligand, and we want to see whether it's if we have an alcohol group on the actual compound, whether that would help the reaction. So that brings me to this last part here, where we want to investigate the Pauson-Khand reaction on an allylic alkene, so with the alcohol in here, and also the enone, where you have an alkene next to a carbonyl group. So in order to make this sort of compounds with the enone in place, there was a bit of challenges as well, but it's a 2-step process, starting from this protected aldehyde. And we were able to vary this R group here by using also a 2-step process to generate this aldehyde here with different R groups, R1 and R2. This intermediate here actually quite susceptible to acid, so we have to avoid acid or chromatography, but using the condition developed by Corey with a buffer with sodium acetate, we were able to get to the desired compound where these carbonyl can be converted into the alkene to get to our intermediate with different functionality of the R1 and R2. However, when we subjected this to the Pauson-Khand reaction, we didn't get our expected product. What I actually isolated was these very high molecular weight compounds, which probably can be attributed to the mass of this sort of dimerization of these two instead of our desired product. So the enone doesn't seem to help. What about the one where we have an allylic alcohol? So as you recall here, the alcohol ligand helps this reaction. So that is why we want to introduce an alcohol group in the actual compound to see whether this extra chelating property would help the reaction. So the allylic alcohol alkene can be generated using this quite simple synthesis. And when we subjected the alcohol unsubstituted, it only gives us the degradation of the complex alkene. Also, when using a literature precedent for compounds with allylic alcohol, it also gave us very small quantity of the isolated ketone instead of the allylic here. And what about under our condition? Unfortunately, it also doesn't give me the desired product for the allylic alcohol. So what we actually isolated out from the reaction using these intermediates is either high molecular weight compounds suggesting a formation of dimerization, or we recovered the alcohol ligand. So this makes me ask a question that possibly the dimerization formation can be decreased by using a more diluted reaction condition, and also if you allow the time for the complex to form with the alcohol ligand, it may help the reaction. So by changing the concentration of the reaction, so it's now 10 times more diluted, on this unsubstituted allylic alcohol, the yield was actually quite good. However, when we move to our trisubstituted alkene compound with the allylic alcohol in place, under the diluted condition, as well as time allowed for this alcohol ligand to form complex, we were able to isolate a small amount of product. So you can see here that there are two factors that may contributing to this reaction: the alcohol ligand and also the ligand, the alcohol that's actually on the compound. So in order to tease out whether it was my alcohol ligand or is it this one here helps the reaction, I tested out without the reaction in the absence of the alcohol ligand. And although I was able to isolate some of the expected product, but this reaction was shown by analysis was very complex, so it's from mixture of products. So to summarize this part, the chelating alcohol seems to help the reaction when you have an electron-rich substituted aromatic ring, and also the low concentration seems to prefer to prevent the formation of dimers. So having made this, having made these precursor using the developed procedure of the Pauson-Khand reaction on a trisubstituted alkene, the last thing that we want to do is to subject it through to get to the final compound, and this work is still in progress. So we propose, this is the synthesis scheme that was proposed that may get us to this final compound as shown here, from using the reduction to form this allylic alcohol, followed by epoxidation to form this three-membered ring with an oxygen here, and then walk this epoxide from this position to this position here, which can be subjected to the addition of an organolithium of this moiety here that can be further cleaved via an ozonolysis to get us to the final compound. However, this work is still under investigation, and I hope that it will get me to the final product. So in conclusion, today I've talked to you about the story of Silvestrol, how the literature has focused on to improve the pharmacokinetic property of this compound by changing the substituents around this ring core. And what my work contribute to this piece of the puzzle is that we show that the C8 methoxy group is important for the activity of Silvestrol, And also, during the course of generating this compound series, we found that we were able to improve the very classic reaction, the Algar-Flynn-Oyamada reaction, by the addition of phase transfer, which allows us to access the key intermediate, which is not able to form using the classical standard condition. And I also talked to you about our effort to try to modify this ring core, and what we were able to achieve from this is we show that we can make different analogues with varying the ring size here, and also an analogous tricyclic without the oxygen replaced by a carbon linker. And during this work, we were able to report the first example of a Pauson-Khand reaction on a tri-substituted alkene, which hasn't been shown before, using alcohol as the promoting ligand. And I was able to investigate the electronic factor on the substituted, on the tri-substituted Pauson-Khand reaction, and which, even though that we weren't able to generate the parent tricycles starting from this enol ether, replacing that with the carbon linker enable us to access the tricycles where this substitution is either tri-substituted or di-substituted. When you move to a more electron-rich system, the allylic alcohol seems to be more favored compared to a more electron-poor enone system. So for future work, to fill in the missing piece of the puzzle, we would hope that we were able to generate the final compound starting from the precursor that I talked to you about, that is accessed from the Pauson-Khand reaction that I have developed. And it would be great to be able to see how it performs in biological assays. So that's the end of my talk, and I would like to thank these wonderful people. Without them, this wouldn't be possible. So firstly, I would like to thank my supervisors, Chris and Guillaume, for their guidance throughout the project, and for this really great project where I can just explore a lot of chemistry. And I would like to thank my committee members, Ben, David, and Paul, for their support and ideas along the way. And I would like to thank the MedChem lab member, especially John, Jack, Bill, Pat, and Brad, for really a lot of support in the chemistry and the discussion and mechanism as well. And I have to thank Wilco, Dana, Louisa, for just me annoying them all the time in the lab since we moved to Parkville, because we just started here and there's not enough chemicals and all that. And also, I thank Jonathan for getting me to WEHI. That's how I started here. And I want to thank my great PhD buddies, Sylvia and Kung, for going through with me with the up and downs of PhD. And for the biology part, I want to thank David, Lisa, and David Segal for testing the compounds. And also, of course, the funding bodies, the APA Scholarship and CTX for Scholarship. And thank you for your attention.
All right, any questions for Duong for a couple of minutes? Really, yes.
You showed at the beginning that Silvestrol was more toxic to B cells and to T cells. Do you have any explanation for that, thinking that it's related to that
The selectivity against B cell, I would think that it's because the downstream effect of Silvestrol has been shown to downregulate MCL1, and so it would be whether the B cell depends on this protein for survival versus the T cell. And also the fact that it's not toxic to, it's less cytotoxic to the normal cell versus the cancer cell suggests that there are also other mechanisms evolved as well. Yeah.
So I also couldn't follow the chemistry that carefully, but you've obviously gone through a very elaborate process to improve the physicochemical properties of the compounds. But I didn't get an impression for whether there were any steps along the way when you were testing whether you were still maintaining the ability to inhibit translation, or whether you basically had to go through this whole process with a kind of, faith that you would ultimately generate something that would inhibit translation.
Unfortunately, we haven't been able to test because I haven't been able to generate the compound that's with all the elaborated substituents like the natural products. But because the current synthesis only allows you to access that core, and because a lot of work has gone through exploring the substituents around the core, and even though they are able to improve some pharmacokinetic properties, but there's still a lot of challenges remain, such as the narrow therapeutic windows. And so we want to explore something that's different, so which is changing the core itself. And because the current synthesis doesn't allow that, so I'm quite happy that with the synthesis that I've developed, we can now make that different core, and it can be extended to other different ring size and all that. And if we can generate the final compound, then we'll be able to find out whether that will affect the compound in inhibiting translation. Yeah.
But I guess a question along those lines is whether there are any studies of how Silvestrol inhibits the complex, whether you've got any structural information about bits of that molecule that you can't afford to change.
The actual binding site of Silvestrol and its target eIF4A is not known. The only hint that we have up to now is that Mutagenesis study, where they show that there are 6 residues on the eIF4A, close to where it binds to the mRNA, that are important for Silvestrol or rocaglates to bind and inhibit translation. But the actual binding mode, we don't know, and that is why I think it's important to explore different parts of the molecule in order to, find out whether that's the case. Yeah.
I'm sorry, I missed the first 10 minutes of your talk, so forgive me if I'm asking you something that you put up. But my question is anything known about the biosynthetic pathway of Silvestrol, and indeed whether any enzymatic steps, for example, in that might be useful in providing a platform for derivatives?
So it's actually interesting. So the approach that we follow at the first part of the synthesis to generate the deletion analog is follow the synthesis developed by Poco's group, and his synthesis is based on the proposed biosynthesis of this class of compound. We did that by chemical reaction and not enzymes. But the important one in that biosynthesis is the presence of an oxygen. So you need that oxygen there in order to, in nature, it does that by a reaction called microaddition. And in a chemical approach that developed by Poco, he needs a cycloaddition reaction that also requires an oxygen. So if you replace that with other atom, that wouldn't work. So I guess with where we are approaching is an alternative approach that can replace that oxygen with other atom to generate a similar compounds, but we're changing the actual core structure. Did I answer your question? Yeah.
I like the look of your big dimer, your eno dimer. Did you get enough to characterize it?
No, it's actually from LCMS because I'm not able to NMR this because it's got cobalt complex in there. So I can only just proposing from the LCMS that this high molecular mass of the one where with the cobalt still on it and the one where it's actually close, but that's just from working through the mass. It's not the, I guess it's just a proposing structure. It's not the actual like full characterization just by an LCMS. Yeah.
Okay, so given the time, I think we'll wind up there, but hope you'll join with me in thanking Duong for a really fascinating talk. Thank you.